brief story about our work, and then Cassie will take over and tell you a bit more about her work. So by way of introduction, cells are very highly organized systems, and so you can see from this slide here that the cell has organized various components in different colors here in different portions of the cell. So they're very highly organized systems, and part of this organization is largely driven by microtubule-based processes, and one of them that I'm showing you here is intracellular transport, which is the movement of materials around inside of cells in order to organize the internal components of those cells. And this great movie that I love to use is actually a slime mold that some researcher found growing on his fish tank and put it under a microscope, and you can see the absolute crazy streaming of materials inside of this mold, and all of that streaming is driven by motor proteins that move along microtubules, which is something that my lab is very interested in at Davis. And so what are microtubules? Microtubules are dynamic polar polymers. We've heard a bit about that from prior talks. And these polymers they underlie cell shape and cell organization. And so as part of the dynamics that I'm showing you here inside of a typical cell that we would look at in the lab, you can see that the microtubules are sort of growing and shrinking in a very dynamic fashion. We call this dynamic instability. And this dynamicity is actually critical for microtubule function in most cells. And part of that function, as I just mentioned on the previous slide, is by allowing the movement of intracellular organelles within these cells and along the microtubule tracts. And so on the right-hand side, you see the red microtubules inside of a living cell. And in green are membrane vesicles that are being transported along those microtubules by motor proteins that, as I said, move along the microtubules, such as cars move along our roads on our highways. And so what is a microtubule itself? Well, microtubules are polymers that are composed of tubulins. These tubulins are diagrammed here. They're alpha and beta tubulin dimer, and they're bound to nucleotides called GTP that can be hydrolyzed into GDP, and that, depending on the type of nucleotide that's bound to the tubulin subunit, it can alter the shape or conformation of that tubulin subunit, which in effect alters the shape or conformation of the entire microtubule lattice, which I think is very important and critical for the questions that people are interested in here about how the microtubule lattice may play a role in consciousness. And so, again, just to highlight the dynamicity of the microtubules, this is a neuron in culture. And what you see here in the little, hopefully you can see the little green dots that are flowing around in that cell. And those green dots are the ends of microtubules that are growing in this cell. And so the neuron itself has many, many dynamic microtubules that are growing and shrinking throughout the entire cell process. And on the right, on the right-hand side there, you could see, yep, I won't play again. But on this side, on the right, you could see the tip of an axon, and hopefully you were able to see that there are many dynamic microtubules even at this higher resolution image, just to highlight the fact that microtubules, again, are constantly growing and being turned over within cells. And I think that's an interesting question for people who are interested in consciousness, is how does this microtubule dynamicity might play a role in the conscious processes that we heard discussed already at this meeting. Okay, so I want to tell you a little bit about the projects that Kassandra and I have worked on related to microtubules, and in particular the microtubule-associated protein called tau, which is a very famous protein because it's implicated in Alzheimer's disease. And so tau was originally identified as a MAP, or a microtubule-associated protein, in 1977, so we've known about it for quite a long time. It became very famous in the 1980s when it was discovered that tau was the primary component of these tangled plaques that are found inside of Alzheimer's disease brains. And despite the fact that since then there's been intensive studies, probably more than 100,000 papers published on tau, we still don't understand what tau does. Why do our brains have tau? Why do we need tau? And what happens when tau goes into this disease state? And so if you Google this, you'll find this picture or a similar picture anywhere that you look. This is the dogma in the field that neurons contain microtubules, as I showed you in those movies just prior. And those microtubules are covered in tau proteins. We believe this is true. But the idea is that in Alzheimer's disease, something goes wrong with the tau, and the tau falls off the microtubules, and then the microtubules become destabilized, and they fall apart. And that's a dogma, but I and Kassandra don't believe that there's a lot of great evidence to underlie this dogma. There's certainly evidence that tau aggregates, as I mentioned. There's not a lot of evidence that the microtubules themselves fall apart, as everyone suggests by this dogma. But that's something that I think requires substantially more investigation by the field. So what we do in our lab is we study microtubules and MAPS and tau in particular in vitro, meaning that we take proteins outside of cells and we have complete control over what we do with those proteins in a test tube. And so what I'm showing you here is a microtubule that we have taken and put it on a glass slide. So there's no cells involved here. It's just a single microtubule. And what we're going to do is we're going to add tau protein that's going to be shown in green. And one thing that we noticed right away when we did this experiment was that tau protein forms very interesting clusters or regions of high density along the microtubule. This is very different than most other MAP proteins that we studied, where the proteins bind sort of uniformly along the microtubule. So what we did is we've studied the formation of these tau envelopes, we call them. We believe that they're enveloping the entire surface of the microtubule. And the references for some of this work that is already published are shown here. So one thing that we can do with our high-powered single molecule microscopes in the lab is actually look at the dynamics of microtubule-associated proteins live. And so what you see here is a microtubule in blue, and in green we've added tau molecules at a concentration where they form these envelopes. So you see that large green cluster or envelope of tau molecules bound to the microtubule. But in red, I hope you can see there are individual dots that are very dynamic, and those are single tau molecules at a low concentration of tau that are shown in red. And what you can see is that the single tau molecules are very dynamic. They bind to the microtubule and they diffuse along the microtubule surface. But once a single red molecule enters into the green tau envelope, I hope you can observe that it becomes trapped or entrenched in that tau envelope. So the dynamics, the molecular dynamics that you're observing live here are actually changed when the tau encounters one of these tau envelopes. And we believe that these tau envelopes represent a oligomerization or a self-association of the tau molecules on the microtubule. And this is really important because self-association of tau was previously thought only to occur in disease, such as Alzheimer's. But we believe it actually occurs on the microtubule in a very physiologically relevant way. So this is direct observation of reversible tau self-association. I underline reversible because, again, in Alzheimer's disease, the tau self-association is thought to be irreversible. That's why you find all these plaques and aggregates that are found in diseased brains. And tau dynamics, the single molecule dynamics, are greatly reduced when tau molecules enter the envelope. Okay. So to go a little bit further into this microtubule lattice state that I mentioned before, the microtubule lattice can exist in at least two different states, which we call expanded, or this occurs when the tubulin dimers are bound to the GTP that I mentioned earlier. And it can also occur in a compacted state, which occurs when the tubulin is bound to GDP. And so this expanded and compacted refers to the spacing between the tubulin dimers within the lattice. And one of the things we noticed is, well, first of all, there's structural biology from other labs that have shown that the spacing changes by a very small amount. It's only about 2 angstrom, which is very, very small at the molecular scale. But nonetheless, one of the things we've noticed during our studies is that these tau proteins actually can induce a change in the lattice structure of the microtubule. And I want to show you this movie here. So here's a movie where we have tau in orange, and it's binding to a microtubule. And I hope what you notice as the movie plays through again is that the microtubule was initially bent, but as the tau formed that envelope, it actually straightened the microtubule. So in other words, by forming that tau envelope, the tau protein binding to the microtubule actually stiffens or alters the conformation of the microtubule. And we have shown through different experiments, such as the one shown here, where we can track individual tubulin molecules within the microtubule lattice. We can see that they change distance over time, and we can measure that they actually compact by about 4% when tau binds to the microtubule lattice. So the take-home message of this is that the microtubule lattice is not a static structure, it's actually a dynamic structure, and that the binding of other molecules, important molecules such as tau, can actually alter that structure of the lattice, which again maybe has implications for things such as consciousness that people are interested in at this conference. So one final thought here about tau function is that tau is known to regulate the function of other microtubule-associated proteins. There are hundreds of proteins that associate with microtubules in cells. And so one of them is called spastin, which is a microtubule severing or cutting enzyme. What you see here is the microtubules in blue that are covered in spastin in green, but the spastin is not doing a whole lot. It's just binding to the microtubule and diffusing around. If I add tau now shown here in red, and I hope you can see those tau envelopes are the dense red regions along a microtubule, you can see that the spastin molecules are now excluded from the tau envelopes, and you see the green accumulates outside of the tau, the red tau. And by excluding and accumulating the spastin in those regions, it actually leads to the spastin to cut and sever the microtubule lattice around the tau envelopes. And so what you're seeing here is that the tau envelopes, by forming an envelope around the microtubule lattice, are dictating where other proteins can associate with the microtubule itself. And we believe this could represent a novel function of tau. As you recall, I mentioned earlier in the talk that people still don't understand what tau is doing inside of live cells. And through data I don't have the time to show you today, that we also believe that tau regulates the movement of motor proteins, as I showed you in the movies earlier. So here's a molecular model of what we believe the tau envelope could look like. In gray is a microtubule lattice, and you see tau shown here in orange. And the structure that is in dark orange, we actually have real data for. That comes from actual structural data. The rest of the molecule, we don't know what it looks like, so it's modeled here as a sort of a unstructured noodle. But we believe these tau envelopes can control which microtubule-associated proteins can access the lattice, and most importantly, the tau envelopes can change the underlying microtubule lattice from expanded to that compacted form that I mentioned earlier. So with that, I want to turn it over to Kassandra, and she'll tell you about her work on tau and organisms.
So when most people think about tau, you're normally thinking about how not necessarily tau on the microtubule, but what happens in disease state. And that's when tau becomes hyperphosphorylated, falls off the microtubule, and self-associates into these neurofibrillary tangles. And this is associated with a number of different neurodegenerative diseases that are often now called tauopathies. And this is just one example here. This is actually a brain slice from a 27-year-old deceased football player who was actually convicted of murder. And you can see the tau inclusions by eye in this brain. And that's insane to think about. When you actually look at the tau molecule, it's 441 amino acids long. That's only 44 kilodaltons. And yet it can self-associate into these massive tangles that you can see by eye in a brain slice. So, like Rick was saying, we have been incredibly interested not only in the physiological function of tau, how it associates with the microtubule and how it self-associates along the microtubule, but how this goes awry in pathology. And so we've been incredibly curious to biochemically dissect how tau self-associates on the microtubule in a reversible manner, but how this ends up getting converted in pathology to an irreversible self-assembly off of the microtubule. And in addition, in addition to doing this biochemically, all in vitro, all in a test tube, we have decided to also use an organism in order to study this. And we found some pretty surprising results that I want to share with you that, and thanks to this conference, have made me think very differently, not only about tau and microtubules, but about overall organism behavior. And so we actually decided to use a Drosophila model. And this is because many labs before us have overexpressed human tau pan-neuronally, just in the entire brain of the Drosophila animal. And what they found, kind of disappointingly, was that there was age-related neurodegeneration. There's just a little bit more neurodegeneration in older flies. So we thought to ourselves, maybe we could do something here to actually induce neurodegeneration at an earlier age, maybe we could induce this tau oligomerization off of the microtubule, and then we could begin to genetically dissect this pathway that leads from molecules of tau normally reversibly self-associating on the microtubule to pathological filaments that are irreversible self-assemblies. And so what we decided to do was subject these flies to blunt force trauma and give them a traumatic brain injury as has been done in previous labs, and this is specifically from the Wasserman lab. So we collect virgin males and virgin females right after eclosion, right after they hatch. We separate them. We need to control as many variables as possible here, and we specifically take the virgin male flies when they are 1 week old, and we subject them to blunt force trauma, which gives them TBI, and I can go into this more later if you're curious about it. 24 hours after this blunt force trauma, we then can analyze these flies in a variety of different ways. We can analyze them biochemically, just grind them up. We can take their brains and analyze their brains via immunohistochemistry, and we've also performed a number of behavioral assays. And what we found, which was rather surprising to us, was that when virgin males are subjected to TBI, they are dramatically more aggressive. So this is a competitive courtship assay, and I actually think this is flipped. My labeling here, I apologize. So the two TBI virgin males are actually on the left-hand side. In a competitive courtship assay, you take 2 virgin males and you put them together with 1 virgin female. And normally there's a little bit of aggression, but when you take these males and you overexpress tau and subject them to TBI, they become incredibly aggressive with one another to the point where they actually don't even initiate copulation with the virgin female. And what we actually end up seeing is that the males that are subjected to TBI exhibit twice as many aggressive acts and spend twice as long in this aggressive behavior, which if you look into any Drosophila literature, this is an incredible increase. And so we're going to be short on time here, so I'll just kind of go through this really briefly. Essentially what we found is that there is a subset of dopaminergic neurons that seems to be responsible for this. So we did a GAL4 driver screen. So when you drive tau expression specifically in this subset of dopaminergic neurons, and then you subject the flies to TBI, we see the same aggressive phenotype where the virgin males are incredibly aggressive with one another and far more caught up in fighting one another than in copulation. And we thought, okay, there could be 2 things that are happening here. Overexpressing tau plus TBI could be leading to degeneration of these neurons, or it could be leading to activation. I thought it was degeneration because tau is associated with neurodegenerative diseases. But we find that if we silence these neurons, the males don't do anything. You silence these dopaminergic neurons and there is just no zest of life. There is no will to live or do anything. This actually correlated with no significant increase in neurodegeneration at this time point. And so we actually don't think we're killing these neurons. In contrast, we actually think we're activating these neurons because when we specifically activate these neurons, we see an increase in aggression. But here's the interesting thing. We never see one particular behavior that we only see when we overexpress human tau. And whether or not we have a traumatic brain injury, we will see this behavior. Oops. Uh-oh. Okay, and this is this behavior here. So these males will go after each other's wings. And I find this to be incredibly interesting because we've never seen this in the control condition. So these males will specifically attack the other wings of these other males. And we find this interesting and particularly insidious because a male, in order to initiate copulation, needs to extend its wing towards the female. That's how the copulation is initiated and courtship is initiated with the female. And yet this male fly that has overexpressed tau and been subjected to blunt force trauma is specifically attacking the wing and therefore the reproductive fitness of the other male. And I find this really interesting because we do not see this when we don't overexpress human tau. So there's something specific here. So what is actually happening? What might be the underlying reason for activation of this particular subset of neurons? Again, we went biochemically, we went back to our biochemistry, and we like to try to work on every single level in order to figure this out. And essentially what we found was that the human 2N4R tau, as well as every other tau isoform that we've looked at, will co-assemble on the microtubule lattice. And so this is what Rick was talking about before. The tau will alter the underlying structure of the microtubule lattice, making it more hospitable for other tau molecules to come and bind in that exact same region. And so when we have two different colors of tau, no matter the isoform, they will always co-assemble. They're highly miscible. However, when you actually look at fly tau and human tau, they actually segregate into these homotypic patches. And what we find is that the human tau will actually outcompete the fly tau. And so there's competition on the microtubule lattice between certain maps. And when we look at the human tau, it's quite distinct from the fly tau. And so we think that when we overexpress the human tau, it may be outcompeting a number of other maps in the neuron, as well as the fly tau. And then the human tau becomes the predominant map that is binding along the microtubule lattice. Well, what effect might this have if you have all of the microtubules coded in human tau now? One effect that we have observed, as well as a few others, but I'm just highlighting this one, is a difference in microtubule dynamics. And so this, again, is a purified system. We can watch microtubule dynamic instability, meaning we can watch microtubules grow and shrink here in blue. This is just tubulin growing off of a seed. It'll grow and shrink and grow and shrink. If we add fly tau to this system, fly tau just stabilizes the microtubule and just allows that microtubule to grow and grow and grow and grow and grow with no catastrophes. If we add human tau to this system, human tau allows for this dynamicity. And we've started studying a few different versions of this, as well as a few oligomers, and the results are quite striking, but I don't have time to go into it right now. The main takeaway from this is that we think that human tau is actually affecting microtubule dynamics within the neuron, and by affecting microtubule dynamics within the neuron, most likely leading to its activation. And there have been a number of papers that we have published, and from other labs as well, such as Peter Boss's lab, that has shown that microtubule dynamicity is incredibly important for neuronal activity, for branch formation, and just for overall dendritic spine maturation. And so tau is really just one of many maps, probably the most disease-relevant one. And we have been interested in a number of these maps, more so than is shown here. What I hope you appreciate from this model is that all of these maps bind in different regions on the microtubule lattice, and they all have really distinct effects on the microtubule lattice. Some of them will prefer a compacted lattice, like tau, and actively compact. Others will actively expand. And so depending on which microtubule-associated protein is interacting with this microtubule, it will cause the underlying lattice state to be in one conformation or another. What we find particularly interesting is how maps have evolved with the complexity of the nervous system. So if you look at a simple nerve net, such as that of Hydra, we have 3 maps that we can find so far. And then when you develop a little bit more complexity into the bilateral nerve cord of worms, for example, you have the extraction of one and the addition of another. When we start to get to flies, we have the expansion of one particular map group called double cortin. And then when we get to mammals, a lot of these maps expand. There's duplications within the genome that have occurred, and MAP1 family has also expanded as well. But what I find really fascinating about this is that 6 of these maps in particular are specific to invertebrates. And then when you actually look at the primate maps, they are very, very distinct. And we believe that we are uncovering a lot of functional differences between these maps, where we think that once we have developed greater complexity within the nervous system, we have specifically evolved particular functions of maps in order to regulate certain microtubule processes and regulate the underlying lattice structure. And so we've been wanting to delve deeper into this now to really understand why it is that we have greatly expanded our map toolbox as we have increased the complexity of our nervous system. And in order to do this, like we had first contacted Stuart, we really want to figure out how different anesthetics and psychedelics affect microtubules, microtubule-associated proteins, and the various microtubule processes that we can really analyze very deeply, both in vitro and also using ex vivo cellular experiments as well as organismal studies. So we're still looking for a funding mechanism for this, but this is something that Rick and I really want to begin to explore more, because we think that if you have particular maps, and here I'm just showing you a map 7 and tau, these are two maps that compete with one another, and we believe that they alter the underlying lattice state depending on which one is bound. Map 7 prefers the expanded state, tau prefers the compacted state. It would be really fascinating to see if any anesthetics or psychedelics begin to favor a particular lattice state or a particular microtubule assembly that will then enable certain maps to bind and perform very specific functions within the neuron. And we actually have very preliminary but recent data on some microtubule dynamics assays in cells. And again, microtubule dynamics are incredibly important, not only for overall spine development, but mainly for spine stability and maturation. So with that, I'd like to thank you all for listening to our split talk. We need to thank the members of our lab. So we run 2 separate labs, but we're one big happy family. And the main people who have worked on the tau projects are Christine, Roylia, Tracy, and Sheng Yu, with Joey pulling up the rear. And all of our funding sources that have contributed to this project thus far. Thank you very much.
Thank you. That was great. I have a question. There's some papers that show that tau actually is a signal, traffic signal for the motor proteins to get off and deliver their cargo for synaptic plasticity. So could they be coding for memory?
We think so, probably. So, so Rick was actually the person who figured out how it is that cytoplasmic dynein became precessive in his landmark science paper in 2014. It seems so old. It seems so long ago now. And so, since then, it's completely opened the field for studying motor proteins and how these maps are affecting these motor proteins. And so, yes, we have a publication, we have multiple publications now analyzing the role of all of these different maps and how they direct and guide motor proteins to specific regions of the cell. And so, yes, that becomes an enormous question, especially with regards to delivery of particular synaptic vesicles, the uptake of synaptic vesicles, the internalization. And so we do think that the maps are guiding the motor proteins and their trafficking.
Part of learning. Any other questions?
It has to be a good one and a short one.
Who? Is that Tam? Shout it out.
No, I get it. Here's a mic real quick.
Yeah, super interesting stuff, and I'm curious, just looking at your videos and what you're talking about in terms of mechanisms, it seems like there's some analogies between myelination of neurons and what's going on here with tau and microtubules. Have you looked at conduction and differences in conduction from that tauization?
Yeah, that's a really good question. We have not done that. There is a colleague. There's a colleague of ours who is looking at microtubule dynamics within glial cells, and specifically with regards to how the myelin sheath is organized in that kind of really beautiful blanketed fashion. We haven't done anything like that. I don't think she's done anything like that yet either. But that's a really good question to begin to maybe analyze that with overexpression knockdown studies. Yeah.
Any other quick ones? One more. Shout it out, please.
Great talk. So my question is, given the effects of tau on the dynamics of microtubules, how do microtubule destabilizing agents affect the association of tau on the microtubule?
So that's a really interesting question. Tau I'm sorry. So I'll call out, we have a colleague, Peter Boss, who's done some really great work in this area. Essentially, tau is not a stabilizing protein that everybody thought it was. There's now a lot of research, both in vivo and in vitro research, that shows that tau is not a stabilizing protein. So when you overexpress tau and you treat with a destabilizing drug, colchicine, for example, the microtubules will still depolymerize. There are other maps that are actually true stabilizers. And we think that depending on the map that is bound, that the microtubule lattice may be stabilized in a very, very particular state. And we think that other maps may be the better stabilizers in this particular area. Map 6, for example, map 7, map 9.
Great. Thank you very much. Awesome.